Hello everyone, welcome to our channel Medico Mentor. Today we are going to dive into type 4 hypersensitivity reactions, also called T cell mediated hypersensitivity or delayed hypersensitivity. But first, let's understand the meaning of hypersensitivity reaction. When your body encounters a foreign antigen, it typically mounts a defense to clear it. In a hypersensitivity reaction, this response is either excessive or inappropriate. Immunologists categorize these reactions into four distinct types, type 1 to type 4, based on the specific immune components involved. Type 1, type 2, and type 3 reactions are primarily mediated by antibodies, whereas type 4 stands apart because it is entirely driven by cellular immunity, specifically our T lymphocytes. So let's break down how this works, why it takes time to develop, and the clinical scenarios where you will encounter it. The first few things that you should know about type 4 hypersensitivity reactions are these reactions are entirely mediated by T lymphocytes, including both CD4 positive T cells and CD8 positive T cells. Second, it doesn't just happen out of nowhere. It develops in response to antigenic exposure in an individual who has been previously sensitized to that specific antigen. The first exposure primes the immune system, subsequent exposures trigger the reaction. And third, unlike the immediate anaphylactic reaction seen in type 1, this reaction is delayed by 48 to 72 hours after exposure. Because of this distinct window, it is universally known as delayed type hypersensitivity. So, where do you see type 4 hypersensitivity reaction in your clinical practice? A few examples include autoimmune diseases such as rheumatoid arthritis and Hashimoto's thyroiditis. These are the conditions where the body mistakenly targets self antigens. Joints in case of rheumatoid arthritis and thyroid in case of Hashimoto's thyroiditis. Similarly, there are a few everyday contact allergies such as poison ivy or nickel allergy coming from jewelry that causes pathological reactions to the environmental chemicals. And there are certain persistent microbes such as Mycobacterium tuberculosis or Mycobacterium leprae, where the immune cells encounters these stubborn intracellular pathogens and it cannot easily clear those, so it deploys chronic type 4 response to clear those organisms. Type 4 hypersensitivity is divided into two distinct pathways depending upon which primary T cells is involved, such as cytokine mediated inflammation, which is driven primarily by CD4 positive T cells. Here, these cells act as generals and they secrete chemical signals to recruit and activate other inflammatory cells. Whereas there is other direct cell toxicity, which is driven by CD8 positive T cells. Here, these cells act as soldiers, directly destroying the target host cells. Let's look at the visual overview of these two processes. In the top panel, you see an antigen presenting cell showing a tissue antigen to CD4 positive T cells. Then T cells releases cytokines, triggering an inflammatory cascade that ultimately causes tissue injury. Whereas in the bottom panel, antigen presenting cells are presenting an antigen to CD8 positive T cells. And instead of just calling for backup, these CD8 positive T cells bind directly to the target cells and execute cell killing and cause direct tissue injury. So let's learn about the CD4 positive pathway first. There are antigen which can be exogenous from environment such as chemicals, or it can be endogenous such as self antigens leading to autoimmunity. So there are naive T cells, CD4 positive T cells for recognizing those antigen, and naive in a is it is called naive in a sense that these cells haven't got encountered with any antigen yet. So naive CD4 positive T cells encounters this tissue antigen, which is presented beautifully on a platter by antigen presenting cells, and depending upon which cytokines has released at that moment of time, it differentiate into two major effector lineages, either Th1 cells or Th17 cells. And once differentiated, these two effector cells use different weapons, such as Th1 cells produce cytokines such as interferon gamma and tumor necrosis factor. Now, if you talk about interferon gamma, it is the single most powerful activator of macrophages. Once these macrophage gets activated, they churn out aggressive substances that destroy intracellular microbes but collaterally cause significant tissue damage as well. Whereas Th17 cells, on the other hand, produce Th interleukin 17, interleukin 22, and various chemokines, and their job is to recruit more neutrophils and monocyte to the site of injury and cause intense inflammation. We will now look at three classical clinical examples of CD4 positive pathway: the tuberculin reaction, chronic granulomatous inflammation, and contact dermatitis. So first, starting with a tuberculin reaction. Tuberculin skin test is a classic diagnostic application of type 4 hypersensitivity. Well, first, we inject purified protein derivatives intradermally into the forearm. If the patient has previously been exposed to TB, their sensitized T cells migrate to the site, they release cytokines and generate an inflammatory response. Because it is also known as delayed reaction, we read it after 48 to 72 hours and measure the hard raised area over there called wheal or induration. If an induration of 15 is noted, then it indicates a positive response. When an intracellular antigen completely resists being destroyed, the body then switches from a temporary defense to a permanent containment strategy known as granulomatous inflammation. As shown in this diagram, there is persistent antigen presenting cell activation, which leads to continuous differentiation into Th1 cells and secretion of interferon gamma. The interferon gamma activates macrophages, and these intensely activated macrophages morph into epithelioid cells, and some of these epithelioid cells will fuse together with each other, forming a granuloma, which is surrounded by a rim of lymphocytes to wall that threat off. In this image, you see a localized red itchy rash near a metal belt buckle. This is likely an environmental reaction to nickel, which is frequently present in the belt buckle. The chemicals acting as an antigen recognized by CD4 positive T cells through presented to them by antigen presenting cells, and they CD4 positive T cells gets activated, they recruit inflammatory cells to the dermis and result in this kind of a distinct localized reaction. Finally, let's look at the second major type of type 4 hypersensitivity, that is CD8 positive T cell mediated cytotoxicity. Unlike the CD4 positive pathway, which relies on inflammation via release of cytokines, here cytotoxic T lymphocytes interact directly with antigen bearing target cells and kill them directly by releasing mediators like porphyrin and granzyme. So we see these mechanisms playing an important role in medical conditions like type 1 diabetes mellitus, where they target and destroy insulin producing beta cells in the pancreas. In case of graft rejection, where host cytotoxic T cells recognize a transplanted organ as a foreign antigen and systematically destroy its tissue. And in case of viral infections as well as in tumor, where this virus infected cells or tumor cells are directly attacked by CD8 positive T cells. To summarize the lecture, type 4 hypersensitivity reaction is a cell mediated response which is driven by CD4 positive and CD8 positive T lymphocytes in a previously sensitized individual with a characteristic delay in response of 48 to 72 hours, thus also called delayed type hypersensitivity reaction. CD4 positive pathway, which is inflammation relating to, which causes inflammation. It is the affected cells are CTH1 cell, which secrete interferon gamma to activate tissue damaging macrophages, while Th17 cells, they recruit neutrophils to the reaction site, and the examples are tuberculin skin test, contact dermatitis, and granulomatous inflammation. Whereas in CD8 positive pathway, the cytotoxic T cells directly destroy the antigen bearing target host cells, for example, in case of type 1 diabetes mellitus, graft rejection, and virus or tumor infected cells destruction. This concludes the lecture on type 4 hypersensitivity reaction. We'll talk about type 1, 2, and 3 in other videos. So, thank you for watching. Please like and subscribe to our channel Medico Mentor. Thank you.